brain has over 200,000 kilometers of blood vessels. A neuron is never more than a few microns away from a blood vessel. Despite this vast vascularization of the brain, getting access to the brain is the last goal or the holy grail of pharmacological and pharmaceutical companies today. So what are the conditions which are due to inability for the brain to remove substances or access of treatments to the brain? If we look at Alzheimer's disease, over 65 million people will be affected by Alzheimer's by 2030. And as you see on this histopathology slide, the black dots are beta amyloid plaques which are unable to be removed across the blood-brain barrier. If we look at cancer, and especially gliomas, patients do not survive after 12 months of diagnosis because the tumors are unable to be treated by chemotherapy drugs or cytotoxic drugs. And this is one of the patients which came into our clinic. And this is a child who fell off a bicycle. And as you can see from traumatic brain injury, by the third week, the majority of his brain is outside its head because the brain has been unable to remove fluid. Luckily, this child survived, but had long-term brain damage. My name is Clyde Hutchinson, and at Junction Therapeutics, we're looking at how we can safely control and repeatedly open and close the blood-brain barrier, allowing treatments in, but also allow pathological molecules out. Our technology is based on natural physiological processes. We look at targeting the junction proteins. These proteins interlock endothelial cells and are regulated circadianly every day. So overnight, they're suppressed, allowing weaknesses and sieve to allow flow in and out of essential nutrients in the brain. Our technology is based on all the problems and all the issues which people have had for siRNA. We have used the problems around bad suppression of proteins and poor targeting to our benefits. By only suppressing the tight-junction proteins by 30%, we can open and close the blood-brain barrier up to 10 kilodaltons for 36 hours and safely close. We're just accentuating what a normal physiological response happens in the body. And here's a testament of how seriously we feel that this science is. We've worked tirelessly for the last 10 years making sure that we've got the science right. In our science publication, we looked at Alzheimer's. And what we found when we injected mice over a year, every 8 weeks, that we were getting a decrease of soluble beta amyloid 140 coming out of the blood-brain barrier. What we also found in this transgenic mouse model was that we were getting increased cognitive ability using the T-maze response. This is a Nature publication, so please don't take this as fake news as from our keynote speech. We also showed that we can use this to treat traumatic brain injury, allowing the flow of fluid out of the brain and increase cognitive ability. We feel our easiest route to the clinic is using a glioma model. Glioma is an orphan disease, and also the ability to get a clear pathway with the FDA has already been proved through our clinical studies. So at the moment, we're submitting an IND for a clinical study of 40 patients in Stanford Hospital. Our preclinical work was done in primates, and as you can see here, we have been able to flood the tumor with chemotoxic drug, which is temozolomide, using our siRNA technology. We have other potential indicators, and we're talking to pharma companies at the moment, looking at p-tau, pathologies, lysosomal metabolic diseases, and also other neurological conditions. This is a safe and tolerable treatment. We have seen no histopathology side effects. We have also not seen any toxicity at a genetic and neurological level. This is based on good science. We're based in Stanford. Ben Gurion as well. And finally, we're looking to currently raise 20 million euros, 10 of which is coming from undiluted sources, and the other 10 we're looking to raise from venture. Thank you very much.
Thank you. Thank you very much, Clyde. Yeah, Jury, ask away.
Thanks for your presentation. So there seem to be a few different applications. You're talking about getting beta amyloid, so proteins out.
Yeah.
You're talking about getting fluid out.
Yeah.
You're also talking about getting drugs in.
Yeah.
Is there one of those, irrespective of the regulatory challenges and this trial design and all of the other reasons that inform your choice, is there one of those that the technology works best for?
I think the Alzheimer stories is fascinating for us. It's a very unpopular story to even say the word amyloid in the building, and I think a couple of people laugh when we say beta amyloid. But because this is an ability to remove toxic pathological molecules from the brain, Alzheimer is a big draw for us. We see the glioma as the most sensible way because an Alzheimer's trial is so difficult, so fraught. And our key point at the moment is we've done 26 primates. That's still not a human. We need to know what this does in a human. Even though all her histology and all her results show that this is completely safe, we still want to see how this reacts in humans. So for us, our key interests are Alzheimer's and p-tau, but also the Labiotech storage disease and metabolic diseases. We see that as a huge unmet need.
So just to understand the mechanism, you effectively open the gates,
Yeah.
and whether something goes in or out depends on the gradient of concentration.
It's really old-fashioned science. It's called high concentration, low concentration. And that's what we find. We find with the beta amyloid, when you've got a high concentration in the brain, it diffuses out.
Okay, thank you.
So actually, I would like if you can comment further a little bit on the mechanism. So is there any selectivity that would be involved? Because if you open, basically, if it's a gradient problem, everything can come out.
So this is our reason why we're using bad siRNA. So bad siRNA only suppresses about 30%, so we can then control the size. So we can control the size of coming in. What we don't want to see is neurotoxic biologics getting into the brain. Because we did that study over a year, and we showed absolutely no any histopathology changes in any organs, we're pretty sure that this is just accentuating something natural which goes on. So this is a way your brain uses to get rid of waste. I think, and it's the way your eye uses to get rid of waste. So if you get a little bit of sleep in your eye, that's one of the mechanisms of how you do that. So this is why we're quite confident this is not toxic, because it's a circadian rhythm proteins, it's something natural, and we're not allowing access to the brain, such as albumin or IgG or IgE.
Thanks for the presentation. So maybe I have two questions, actually. Maybe first, the animal model. You mentioned it was the transgenic model.
Yes.
Can you shortly comment on that? And then maybe afterwards, the treatment span. How would you expect it to look in humans?
Yeah, so we are currently repeating the mice trial in primates just to see if the effect we had is because you had a superloaded brain of amyloids using a transgenic model. So we understand that, and that's why before we go into an Alzheimer trial, we want to see what happens in primates. Our primate study, which we've done before, has been a glioma study, and so we know pretty sure that the effect we're getting is a natural response, and it's not something which is due to some blip with a murine model.
Can you elaborate a little bit on the glioblastoma study that you plan to conduct? So
I'll go back a slide. Actually, I'll use this one. So the study we're doing it in Stanford. In the corner there, you see Professor Jerry Grant, who is the head of neuro-oncology. We're doing it in Stanford because we have access to patients. We can confidently have 40 patients within 18 months within the trial. they've been identified, and that's why we're in the process of hopefully starting the trial at the end of this year, depending on funding. We already have sufficient funding from undiluted sources to submit our IND. So we'll get to an IND. We're looking for funding then to do the trial. So the reason we picked Stanford also is that Stanford biopsies every tumor, so then we can look at treatment response as well as so we can do a phase 1, phase 2, so we can look at are we getting access to the brain from our treatment, and also are we getting access and allowing temozolomide into the tumor.
And in terms of business model for the company, are you more, what's the vision of company to have a kind of drug delivery intelligent business model, or more standalone development plans?
So we desperately want to work with pharma companies because we want to see how having our treatment with a combinational therapy, how does that work, and how do we can use this to help, say, in lysosomal storage diseases, how can we allow drugs access to the brain, but also allow it, as we call it, a neurological tap, allowing waste substances to leave the brain safely. And that's our goal, is to partner with pharma companies, really to look at their treatments in Alzheimer's, p-tau, and in lysosomal storage diseases.
Can I just pick up on two things he said? One was that this is a circadian
Yes.
feature. So if this is happening, for example, at night, why don't you just deliver the drug at night?
Because you're not getting, we're accentuating, so the Claudin-5 and occludin are overexpressed in the brain and in the retina blood-brain barrier. So because we're suppressing them further by 30%, we're opening up the interlocution a little bit more. What we don't want to do, some of the other blood-brain barrier technologies, rupture the blood-brain barrier, which you're getting inflammation responses and serious complications. What we're looking at is how can we do this safely, and also how can we do this as a repeated treatment, because it's going back to your point. If you're giving this as an Alzheimer's treatment, you're going to have to give this over a long period of time. So we want to make sure that extensive treatment, say every 8 weeks, is safe and it's not toxic, and we're doing something which is a normal physiological response.
Thanks. So you increase the magnitude of what's happening.
Yeah. We're only opening the gates further. What happens naturally overnight for your brain to release. So for us, it's been important that we get the science right, and that's why we've published in Science and Nature and etc. Because we want to make sure that we do proper studies, because this is such a controversial area. And also, you're opening and closing the brain, so we don't want to cause serious damage to patients. So we want to make sure that this is safe.
And the other question was just on ophtha. So you mentioned there might be an application in the eye. Are you developing that?
We're focusing on the brain, but we have data on the effectiveness of this in easing the effects of glaucoma as well.
Okay, thank you for those questions to jury. Now, any questions from the public? One over there. Just wait for the microphone really quick.
Hi, I'm Ajay. A couple of questions. Do you need a license for the siRNA, and how are you just using naked siRNA as a delivery, or are you packaging it in liposomes?
It's interesting enough, the person who developed it is the packaging is sitting just in row 4 there. So we have a draft license agreement with this, but their patent is running out next year. So the initial conversations we've had going back a number of years, there's been no issue around the use of RNAi technology. Secondly, we use polyethylene as a delivery mechanism. That has already been FDA approved. So we're not trying to reinvent the wheel by going through an extensive regulatory response by using AAV vectors or such, because we don't need to do that, because we're not targeting a gene for therapeutic. we're using this as a mechanism just to suppress it enough to open it up. So it's looking at siRNA at a different level and not looking at it as a level of we need to completely suppress a protein or a gene. We're looking at this as using it as a tool to gain access to the brain. So it's a different way of thinking, and it's taken a while for people to get that view of we're not looking to completely suppress it, and we can deliver this systemically. We don't need stereotaxic injections into the brain. We can use this peripheral injection.
Okay, thank you. We actually don't have time for another question. We have to get to the next contestant. Thank you very much, Clyde. A round of applause for Clyde.